Hey guys, welcome back. So today, as we're looking, chapter 1, which was cell structure. So we are going to continue with this part. So in the last video we actually defined what a cell was, and we learned about the two types of microscopes, which was the light microscope and the electron microscope. So today we are going to see the cell structure. So at first we are going to see the structure of a typical animal cell, then we are going to look at the typical plant cell. So let's see the structure of a typical animal cell. Okay, let's see how it looks like. Okay, so in animal cells, so we just start off by drawing a round circle, so just like all of this. So one of the largest organelle inside the cell we need to know is the nucleus, but in our A levels we actually have to learn and see the nucleus in very detail. So at first we need to draw the nuclear envelope. Okay, the nucleus is kind of situated in the center of an animal cell. So let's see. So this is the nuclear envelope which I'm drawing. So yeah, so this is our nuclear envelope. Inside the nucleus there is a nucleolus. So this is actually a nucleolus. Okay, this actually synthesizes ribosomes or rRNA. Okay, we're going to see those, and this is our nucleoplasm. So let's start labeling. So this is our nuclear envelope, this is our nuclear pore, this is the nucleoplasm, this is the nucleolus, and these are actually the chromatin fiber. Okay, I have almost occupied a lot of space just doing that. Anyways, so let's continue. So along with the nuclear envelope is actually connected to another organelle, which is actually our endoplasmic reticulum. Okay, so let's draw the endoplasmic reticulum. So these ones which I'm drawing right now are actually our endoplasmic reticulum. So you're also going to find out there are actually two types of endoplasmic reticulum. One are the rough endoplasmic reticulums, and the other ones are the smooth endoplasmic reticulums. Okay, anyways, so yeah, so this is one set of endoplasmic reticulum arranged out from there. So this is another endoplasmic reticulum which we're drawing. Okay, so yeah, so these are actually our endoplasmic reticulums. So if we have to make it a rough endoplasmic reticulum, then it will have ribosomes attached to it. Okay, so these circles which I'm drawing are actually the ribosomes which are attached to the endoplasmic reticulum. So these ribosome attached endoplasmic reticulums are actually called our rough endoplasmic reticulum. So this is our rough ER. ER is a short form of endoplasmic reticulum. Okay, now okay, let's draw some ribosomes. So we may also see some endoplasmic reticulums without ribosomes, such as this one. So this is called a smooth endoplasmic reticulum. So the easier way to think about this is that ribosomes actually make the surface very hard. It's no longer smooth. So this rough surface, because of this rough surface due to ribosomes, it's called the rough ER or rough endoplasmic reticulum. And since there is no ribosomes in this specific endoplasmic reticulum, it's called smooth endoplasmic reticulum because it's actually a smooth surface. Okay, now let's see the other organelles. So next we have other something we know, the powerhouse of a cell, the basic structure. This is our mitochondria. So mitochondria. So inside the mitochondria we all know these enfoldings or these folds inside the mitochondria are called the cristae. Okay, so we're going to learn about them later. We are going to explain in details every single organelle of this chapter, no worries. Okay, so next let's do the Golgi body. So Golgi body's shape is like this. It usually faces the membrane away from the nucleus and toward the membrane. This is usually how Golgi bodies usually appear to be. So these are called the Golgi body. Okay, sometimes there are larger ones. These circular ones are actually called the Golgi vesicles. Golgi vesicles. Okay, So sorry, I do not have enough space to write. I'm extremely sorry for that. But they're called Golgi vesicles, as you can see. Okay, next we know this surface, this entire thing is the cytoplasm of the cell. So this is of course the cytoplasm of the cell. Next we have got certain structures, the certain other circular structure, which are actually the lysosomes. So these actually contain enzymes which involve bacteria, helps in phagocytosis. We're going to learn that in immune system chapter in more and in much further detail. Next, sometimes these cells also may have extensions like this, which are actually called microvilli. So these extensions are called microvilli. And what else are there? So these are like the basic structures which we have. So once we go into further details in this chapter, we are going to see more structures which are visible within an animal cell. Okay, there are also centrioles and centrosomes, so we are going to cover that a bit later. Usually there are also exoskeletons, microtubules, microfilaments like this roaming around inside the cell. We're going to learn that in further detail when we talk about cell division, because they actually play a major role in these aspects or in these functions of the cell. So we're going to also cover those. Okay, so often there I missed one important, very important organelle. So there are 3 even smaller circles which are ribosomes. As I already mentioned, on the surface of endoplasmic reticulum I also mentioned, so there are ribosomes. So another thing we need to remember, whenever we are drawing ribosomes in animal or plant cell, they are always 80S ribosomes, because these are eukaryotic cells. So eukaryotic cells are the more recent kind of cells, the more advanced kind of cells, which has 80S ribosomes. And usually prokaryotic cells have a 70S ribosomes in them. Okay, so that's also something we have to remember. Okay, so this is the structure of a typical animal cell. Now, if you look at a plant cell is kind of similar. Plant cell can be of a hexagonal structure, or it can sometimes be rectangular in shape. So rectangular, we've seen palisade mesophyll cells. If you've read in transporting plants and olives, we have seen those cells which are actually rectangular, cylindrical in shape. Okay, anyway, so in plant cells the major difference is that they have a large central vacuole and they have chloroplast. Okay, so they have chloroplast like this. These are the kind of chloroplast. So all the other organelles are basically the same, except they have this chloroplast and a very large central vacuole, and they have this chloroplast, this outer membrane of the vacuole. You need to know the name. That's called the Tonoplast membrane, or you can just simply call it Tonoplast. Okay, and they have a cell wall on the outer surface. They do not only have the membrane, but they also have an extra layer which is called the cell wall. So we are also going to know about the details of cell wall. We're going to know about the primary, secondary, and the middle lamella of the cell wall. So those are the 3 parts of the cell wall which we're going to discuss later. In fact, we're going to discuss every single organelles in detail in the later videos. So these are the basic difference between a plant and an animal cell. And another difference which you can say is that they do not have centrioles. They do not have centrioles. These are for plant cells. Okay, so the major difference is that they have a large central vacuole. They have chloroplast, which consists of chlorophyll, and this is a green pigment which helps in photosynthesis. They have cell walls, which is made of cellulose, and they have a large vacuole, and the outer membrane of the vacuole is called the tonoplast membrane. So these are the basic structures we saw of these different organelles of plant and animal cell. So in the later videos, we are going to actually discuss specific details and functions of every single organelle and their structure. So stay tuned for that. Thanks for watching.